Last-minute studying speedrun because my brain refuses to cooperate under pressure. Let's talk about genetic engineering before I forget everything. Genetic engineering is basically when scientists deliberately change an organism's DNA using biotechnology. Not like traditional breeding, where you wait generations and hope for the best, no. This is direct, targeted surgical gene editing. And yes, genes from one organism can work in another. Why? Because DNA is universal. A gene is a gene. If you transfer it properly, it can function in a totally different organism. Wild. Now enter recombinant DNA technology. Recombinant DNA is DNA made by combining genetic material from different sources, different species, different origins, one DNA molecule. Why do we even do this? To make important medicines, improve crops and livestock, and to study how genes actually work. Now let's talk about the tools, because this is where it sounds scary, but isn't. First, restriction enzymes, also called restriction endonucleases. These are basically molecular scissors. They cut DNA at very specific sequences. Next, DNA ligase. This is the glue. It joins DNA fragments together and seals the sugar phosphate backbone. Then we have vectors. These are carriers that bring the foreign DNA into a host cell. The most common ones? Plasmin's little circular DNA in bacteria and viruses, especially in gene therapy. Now the process, lock in. Step 1. Isolate the gene of interest. Step 2. Cut the DNA using restriction enzymes. Step 3. Insert the gene into a vector using DNA ligase. Step 4. Transfer that recombinant DNA into a host cell. Step 5. Select and express the desired gene. Every step has to be precise or the whole thing fails. No pressure. Applications? Oh, there are many. In medicine, we use bacteria to produce human insulin, develop vaccines, and even do gene therapy. In agriculture, we make GM crops that resist pests and diseases, and crops with better nutrition, hello golden rice. In industry and the environment, we use engineered microbes to produce enzymes and clean up pollution through bioremediation. Now let's clarify GMOs. A GMO is any organism whose genetic material has been altered using genetic engineering. That includes bacteria, plants, and animals. And yes, they are everywhere in agriculture, medicine, and research. Advantages? More food, better quality. Disease treatment. And it's faster and more precise than traditional breeding. But there are concerns, environmental effects, health risks, ethical issues about manipulating life, and unequal access to the technology. That's why ethical guidelines and regulations are needed. And boom, genetic engineering explained. If I forget this during the exam, at least I tried.